Hello and welcome back to AP Bio with AP Playground. Today we're covering topic 3.5, cellular respiration. Just like 3.4, we've got another long lesson here, but we'll be looking at the components of the mitochondria, the steps of cellular respiration, including glycolysis, Krebs cycle, and oxidative phosphorylation, and we'll also be taking a look at fermentation in the end. The suggested skill is representing and describing data. For A, construct a graph to represent the data, including XY graphs, bar, histogram, line, log scale, dual Y, scatter plot, box and whisker plot, and pie chart. The graph should include the following components: the type of graph appropriate for the data, axis labeling, including appropriate units and legend, scaling, accurately plotted data, including error bars when appropriate, and trend line when appropriate. Now, cellular respiration is one of the concepts that on the test you will have to most likely graph, so it's good to have a good understanding of the subject matter. Let's go. First up, what is cellular respiration? So cellular respiration is the way living organisms gain energy by breaking apart biological macromolecules such as glucose. Now, the molecule doesn't have to be glucose, but we will generally be looking at the breakdown of glucose. But other biological macromolecules and other kinds of food either have different pathways or enter the cellular respiration pathway at a different place, allowing them to be broken down and for the body to gain energy from them. But for all intents and purposes of this course, we will be looking specifically at glucose. And there are two catabolic pathways used. The first one is fermentation, which is a partial breakdown of sugars and does not use oxygen, compared to cellular aerobic respiration, which uses oxygen. That makes it aerobic, and it turns macromolecules into ATP. And obviously the primary macromolecule is glucose. Now, fermentation will also generate ATP, but generally not as much. And the exergonic release of energy from glucose is used to phosphorylate ADP to turn it into ATP. Like we talked about, this is an energy coupling reaction. The breakdown of the biological macromolecule is going to form ATP, and the ATP is then able to be used for other cellular processes. And just like photosynthesis, it's going to be a redox reaction, a reduction-oxidation reaction. And taking a look at the equation of cellular respiration, we see we have glucose plus oxygen is going to yield carbon dioxide plus water plus chemical energy. And if you recall, this is the exact opposite of photosynthesis, which took carbon dioxide and water to form glucose and oxygen. So this is another example of energy coupling where the reactants for one of these reactions are going to be the product of the other. And chemical energy is generally going to be 686 kilocalories per mole of glucose. And the way it's a reduction-oxidation reaction, again, by the gaining and losing of electrons depending on the reactant in the reaction. First up, glycolysis. This is going to be our first step of cellular respiration, and it's also used in fermentation. It occurs in the cytosol. So before we enter any organelles of the cell, and both plant and animal cells perform cellular respiration, we're going to be starting in the cytosol. And it's anaerobic. So it doesn't need an organelle and it doesn't need oxygen. And it's really the first evolution. It evolved first as a way to gain energy from biological macromolecules before there was oxygen in the atmosphere. Through photosynthesis, before the mitochondria had joined the eukaryotic cell to perform aerobic respiration. And it requires an input of energy, and you gain energy from it. So you have a net gain of 2 ATP because it takes 2 and you gain 4. So it's net 2. Additionally, it fills 2 NAD+ electron carriers by reducing them, of course, adding electrons to NADH. Now, it's important to not confuse this with NADPH, which is the electron carrier used in photosynthesis. Lastly, it's going to turn a 6-carbon glucose, because of course glucose has 6 carbons, C6H12O6, into 2 3-carbon pyruvate molecules. So if we take a look here at the steps of glycolysis, there's going to be an energy investment phase where 2 ATP are used up and turned into ADP. And then there's an energy generation phase where 2 ATP are gained, 2 NADHs are gained, and 2 pyruvates are gained. The 6 carbon glucose that we've had has split up into 2 3-carbon molecules. And there's an enzyme known as phosphofructokinase, which is an allosteric regulator of glycolysis. Essentially, once enough pyruvate is in the cytosol, it will turn off the glycolysis production pathway, an example of feedback inhibition. So our total production is 2 pyruvate molecules, 2 ATP, and 2 NADH. Moving along to the link reaction and Krebs cycle. Now, the link reaction might not necessarily be considered a separate step, but it is an intermediate step that needs to be addressed when talking about moving finally into the mitochondria. So pyruvate is moved through the membrane of the mitochondria into the matrix, which, if you remember, is the interior space of the mitochondria, just like the stroma is the interior space of the chloroplast. And from each pyruvate, a carbon dioxide molecule is removed, and electrons are used to create more NADH. And lastly, the remaining 2-carbon structure become acetyl coenzyme A. Because of course, each pyruvate, and there has 3 carbons, becomes an acetyl coenzyme A with 2 carbons once 1 carbon dioxide atom has been sent out of the cell. And these acetyl coenzyme A molecules enter the Krebs cycle, where breakdown is complete by turning each acetyl coenzyme A into carbon dioxide. So eventually, through the link reaction and glycolysis and the Krebs cycle, all 6 molecules of carbon that started as glucose are going to end up exiting the cell as carbon dioxide. That's what we exhale. And it takes 2 turns for 1 molecule of glucose because, of course, there are 2 acetyl coenzyme A's that need to move through the cycle. And we'll look at the diagram in a second. And more NAD plus become reduced to NADH, as well as a second electron carrier FAD become reduced to FADH2. So our total production is going to be 4 carbon dioxide atoms, 6 NADH, and 2 FADH2, as well as 2 ATP. And adding that to the products from the link reaction, we're going to have a total of 6 carbon dioxide for all of our glucose molecules, 8 NADH, electron carriers, and 2 FADH2 molecules. And a good way to remember this is NAD and FAD. Those are going to be our 2 electron carriers for cellular respiration, while of course photosynthesis has NADP+. Taking a look here at the link reaction and Krebs cycle in diagram form, we start with our 3-carbon pyruvate, and 1 carbon dioxide has left. NAD has been reduced to NADH, and we now have acetyl coenzyme A, which enters the Krebs cycle, otherwise known as the citric acid cycle because it joins a 4-carbon compound known as citric acid. And this is going to form a 6-carbon molecule, which is going to reduce more NAD to form NADH, and the 2 carbons that came from the acetyl coenzyme A are going to be expelled. Then FAD is going to become FADH2, and lastly, a few ATPs are produced. And we're left back with this citric acid molecule of 4 carbons, which can then bind to the second acetyl coenzyme A and perform another spin of the cycle, removing all of the initial carbons of glucose as carbon dioxide. So by the end of the Krebs cycle, all 6 carbons from the glucose have been released as carbon dioxide. However, we have not really made that much energy, only about 4 ATP or so. So now it's time to learn about the last part of cellular respiration, the part where all of the ATP is going to come from. This last step is known as oxidative phosphorylation, and it's broken down into 2 parts. Oxidative meaning uses oxygen, and phosphorylation meaning it's going to phosphorylate ADP. So we're now in the inner membrane of the mitochondria, and remember the mitochondria is a double-membrane organelle through the theory of endosymbiosis, where it was taken up by a eukaryotic cell, which means that we have 2 membranes, and on the inner membrane of the mitochondria there's an electron transport chain, just like in the thylakoid membranes of the chloroplast in photosynthesis. And again, this is going to consist of transmembrane proteins. And what does the electron transport chain do? It takes electrons from our lovely electron carriers, both NADH and FADH2, and these electrons are going to travel along the chain through each transmembrane protein, and this energy is going to pump hydrogen ions across the membrane from the matrix of the mitochondria into the intermembrane space between the outer and inner mitochondrial membrane. And electrons are moving through this electron transport chain because they are attracted to the final electron acceptor, the electronegative oxygen molecule. And this combination of the electrons plus the oxygen plus a couple more hydrogen atoms is going to form water, which is why water is a product of cellular respiration, and why oxygen is necessary for cellular respiration. If you do not have oxygen, you're not able to power the electron transport chain. The electrons do not have a final electron acceptor that is electronegative, that they are attracted to, that they are traveling towards, and will not pump any hydrogens into the intermembrane space. Now let's take a look and see what happens once all of the hydrogens have been pumped into the intermembrane space. Now we do what's called chemiosmosis. The hydrogen ions in the space form an electrochemical gradient, meaning that both there are going to be more hydrogen atoms in the intermembrane space than the matrix, so they want to move down, but also the charge is going to be different because of all the positivity of the hydrogen ions. But they can't cross the membrane because they are ions and require a membrane protein to do so, even if they want to follow the concentration gradient. And the only membrane protein that they can go through there is ATP synthase. And as protons travel through ATP synthase, as shown here, we're going to get the phosphorylation of ADP and create ATP. So oxidative phosphorylation is the combination of the ETC, which takes electrons from our electron carriers and uses this energy of the electrons moving along the chain to pump hydrogen ions into the intermembrane space, and then chemiosmosis, where these hydrogen ions are going to power ATP synthase, creating ATP. And this total production is going to result in 34 ATP and 6 water molecules. And as you can see, this is going to be the place where we get the majority of the ATP. We only got a few in glycolysis and from the Krebs cycle, but here in oxidative phosphorylation, we've made 34. So, an overview of cellular respiration. We took our glucose and we took oxygen for oxidative phosphorylation, and we made carbon dioxide, water, and chemical energy in the form of ATP. Total production is about 38 ATP, and it ranges from 30 to 38. Different sources say different things, but generally you're going to be in the 36 to 38 range. Now looking at it as a whole, in glycolysis, we broke glucose into 2 pyruvate molecules, we made some NADH, and we produced 2 ATPs at a substrate level, meaning before we've entered the oxidative phosphorylation, before it's the aerobic steps. Next, in the link reaction, we formed acetyl coenzyme A, some more NADH, and we got rid of some carbon dioxide molecules. Now, acetyl-CoA enters the citric acid or Krebs cycle in the mitochondrial matrix, and this is going to get rid of the rest of the carbon molecules of glucose, turning them into carbon dioxide. It's going to produce 2 more substrate level ATP, and it's going to fill electron carriers NADH and FADH and squared, and this is going to result in our NAD and FAD donating electrons to oxidative phosphorylation, where the electron transport chain will power the pumping of hydrogen ions, which will then come back down into the matrix through ATP synthase, powering the synthesis of 34 oxidatively formed ATP molecules. Wow, that was a lot. Now, comparing cellular respiration to photosynthesis, you can see that, first of all, the equations are inverse. Cellular respiration produces water through the binding of the electrons to the final electron acceptor of oxygen, as well as carbon dioxide from the removal of all the carbons in glucose. And these are going to be the reactants used by the chloroplast in order to create glucose. The carbons are used for the glucose, and the electrons from the water in order to replace electrons lost in photosystem 2. And photosynthesis is going to produce the oxygen needed in the final electron acceptor portion of oxidative phosphorylation, as well as the glucose that is broken up by the mitochondria. And as you can see, this follows the first law of thermodynamics. Energy is neither created or destroyed, just light energy entered the chloroplast and went through cellular respiration to become chemical energy that the cell could use. One produces oxygen, one consumes it, one produces carbon dioxide, and one consumes it. Lastly, we need to talk about anaerobic respiration, otherwise known as fermentation. This is the respiration used before the atmosphere had oxygen in it and before the atmosphere was created, and it's also used just when you run out of oxygen for aerobic respiration. And fermentation is going to use the early evolved anaerobic process of glycolysis, which is going to give a little bit of energy. Remember, net gain of 2 ATP, but not that much. But it's better than nothing. The only issue is that the NAD+ carriers are going to get filled with electrons, and once all NAD+ carriers have been filled up, glycolysis isn't able to repeat. And there are two types of fermentation that deal with this issue. First of all, alcohol fermentation. The pyruvate producing glycolysis is converted to the 2-carbon alcohol ethanol, carbon dioxide will be released, and NADH will be oxidized, allowing NAD+ to then be reused in glycolysis. And an example of alcohol fermentation is yeast. There is also lactic acid fermentation, which forms lactate as a waste product and again oxidizes NADH, turning it back into NAD+ to be used in glycolysis. And again, the goal of fermentation is to free up those NAD+ carriers so they can be reused and you can get your marginal 2 ATP from glycolysis repeating. And lactic acid fermentation is also going to be used in humans once they run out of breath. For instance, when humans are running and they start cramping, that means there is a buildup of lactate in their body as a result of lactic acid fermentation. And so here we have some examples. From lactic acid fermentation, we get kefir and fermented vegetables, and in alcoholic fermentation, sourdough, powered by the yeast, and also beer. So, there we go, cellular respiration. Gotta know the equation of glucose plus oxygen is going to yield carbon dioxide, water, and chemical energy in the form of ATP. Starting with glycolysis, where the 6-carbon sugar is going to be split into 2 3-carbon pyruvate, and some NAD+ electron carriers are going to be filled, as well as the gaining of 2 ATP. Then the Krebs cycle, where all of these carbons are going to be expelled as carbon dioxide, some more ATP will be gained, and electron carriers NAD+ and FADH are filled. And lastly, oxidative phosphorylation, where these electrons are going to be used in the electron transport chain, traveling to their final acceptor oxygen and forming water, and this energy of the electron movement is going to be used to pump up hydrogen atoms into the intermembrane space, which will then come back into the matrix through ATP synthase, phosphorylating ADP and producing the majority of ATP of cellular respiration. Lastly, we've got fermentation, which is the anaerobic variety, no water or no oxygen needed, and the only goal here is to be able to reuse NAD+ so that glycolysis can continue. We're almost done with Unit 3, one lesson to go.